Let me record. Okay, thank you. So while we're waiting for Mr. Dashaba, I also want to recognize the presence of Professor T. Fawale. Uh, you're welcome, sir. I'm also recognizing our speakers. They are already in platform. When the time comes, we will try introduce them properly. As well as members of the members of the All Farmers Association of Nigeria. I've seen some of them here already on the platform. Will be recognize you properly when the investor properly. Then, so Mr. Dashaba, are you ready, sir? Okay. Then, if that is not ready, we can move to the Google message from the president of the National Plant Protection Society, National Society for Plant Protection, in person of Professor A.U. Yusuf. Sir, can you take the look? You can almost yourself, sir.
Oh my God.
Okay, Dr. Kazim, while the president is coming on, I think it's also important we recognize the presence of the immediate past Deputy Vice Chancellor, National Open University of Nigeria, Professor Olani. She's here with us, and the National Secretary of the Nigerian Society for Plant Protection, Dr. Orisajo, is also in this meeting. I just thought I should quickly make recognized those
It's very
personalities.
important,
Thank
sir.
you.
Thank you, sir. So the national president, sir. Can you unmute yourself, sir? Dr. Chris, can you assist me in that?
Okay, yeah. Maybe it's a network problem, but the national secretary is here of NSPP. He may want to just chip in a word
Okay,
on
sir.
behalf of our boss. National Secretary, sir, Dr. Risajo.
Thank you, sir. Dr. Sajo, can you unmute yourself, sir?
I hope the unmuting is not disabled from their own end as well. I hope participants can unmute
They can.
themselves. Okay. Yeah, but we can already go ahead, sir, so that we don't spend time. It could also be multiple engagements going on from the other end.
Okay, sir. We will give them some time when they come on, when they. So,
Yeah.
we're having the keynote presenters present here. Dr. Eche will give the Dr. Eche will give the biodata of the first presenter in person of Dr. Diane Mustet from Stellenbosch University, while I try to put up profile up. So, come in. Yeah. So, Dr. Chris, the floor is for you to you.
Okay. Thank you very much, Dr. Kazim. Again, good morning, everyone. My name is Chris Eche, and I am delighted to give here this morning the presentation, or rather the citation of Dr. Diane Mustat before her presentation comes up. So Dr. Diane is a renowned plant pathologist. She's a researcher and also a lecturer in the Department of Plant Pathology at Stellenbosch University, South Africa. And of course, the university is recognized worldwide as one of the centers of excellence for Fusarium tropical race 4 diagnosis. Our interest covers all aspects related to population biology, diversity, evolutionary biology of fungal plant pathogens, as well as the development of molecular markers for pathogen detection and identification. She has proven she has a proven track record in research, teaching, and international collaboration, and is a leading authority in fusarium weeds management.
Go on, sir.
Okay, all right, we had a little hitch there. Dr. Diane is actively involved in the training of postgraduate students and carrying out of sensitization for government bodies and also, as well as farmers. And she has supervised over 29 undergraduate MSc and PhD students with several ongoing and has hosted 4 international internships. She'll discuss with us today on the banana fusarium with TR4, looking at the impacts, races, and symptoms, and diagnosis, and lots more. May I present to us at this point Dr. Diane Mustat to lead us in this conversation. Over to you, Doc.
Thank you so much for the nice introduction, and I just want to say thank you for the opportunity to present today. It's my great pleasure. I'm going to share. You can see my. I think you need to stop sharing.
Okay, okay, I'm coming. I'm trying to go back.
My presentation.
Okay, we can see it. We can see your presentation.
Okay. Telling you a bit about the very devastating banana pathogen. I'll give you some perspective challenges that they. So Fusarium wilt managing plant pathogens in history by soil-borne pathogen Fusarium oxysporum forma specialis cubensis, or FOC, as I will be referring to it from now on. The problem with this disease is that the fungus can actually survive for decades in the soil as durable survival structures, which is called the chlamydospores. And once it's introduced, it's almost impossible to eradicate it. So that's why such days as today is very important, so that we can make everyone aware of the disease to prevent its introduction. We don't have very accurate information on the impact of tropical race 4, but there has been some estimation. So if TR4 risk of spread scale has been estimated that it could affect like 1.65 million hectares by 2014, which has a value of over 10 billion US dollars. When it was first introduced into Africa and Mozambique, there was devastating losses of 15,000 per week, US amount of 236,000. And it ultimately ruined an $80 million investment. As we don't know what is the vulnerability of our cooking bananas as well as plantains, because we haven't really screened it, and I will talk about it a bit later, is then the fungus could, if it's affecting these varieties, it could threaten staple food valued at 4.3 billion US dollars. That's the value of cooking bananas on the continent, and it could have the probability to affect the livelihoods of over 100 million people. So the fungus persist, or there's different strains of the fungus that is identified, and then it's classified into races. And each race can affect a certain group of specific cultivars. So the first race of the pathogen that was caused, or the first epidemic of the pathogen was caused by race 1, where it caused severe losses of Gros Michel in Central America. And during that time, they actually moved the production of Gros Michel to West Africa. And also moved the pathogen. So that's one of the first times that Fusarium wilt was introduced into Africa. But now currently race 1 and 2 is very widespread. As you can see, in orange is basically occurring everywhere where bananas are grown. And then we have race 4 that can affect Cavendish, but it can also affect varieties that's traditionally affected by race 1 and 2, which makes it very devastating. And TR4 was first introduced into Mozambique. I'll talk a bit about that right now after this. And it's only been found there, and also more recently in 2 of the Comorian islands. And in South Africa, in subtropical regions, we also have a different strain that we call subtropical race 4, but I won't focus too much today on that. So Fusarium wilt TR4, the one affecting Cavendish, was first introduced into Mozambique in 2013, and where it destroyed over 1,500 hectares in 5 years. It was introduced into a property that was intended for export. So they really wanted to develop this property to export a lot of bananas to the Middle East. But that investment was completely destroyed with the introduction of Fusarium wilt. On the right-hand side, you can see the cases and how it exponentially went up. Ultimately, it completely destroyed the whole plantation. After severe flooding, as you can see on the bottom there, the cases just went up exponentially because the fungus can be spread. It's a soil-borne pathogen, as I've mentioned, but it can also be spread passively when there's floods on tools, anything that comes first restricted only to Mozambique from 2013, but in 2019 we also found it in an overseas department of France, the island of Mayotte, and we were involved in confirming the first case of TR4. And here you can see how they've done surveillance in Mayotte, and it's quite widespread currently. Then most recently, if you look at the picture here, you'll see, let me just put my laser pointer. This is the island where we found it first, and that's an overseas department of France. But in after we had some training where we trained Mustalifa Mamadi. He went back and did some surveillance in Comores, and we also confirmed it then with molecular testing and VCGs, as well as pathogenicity testing, that it also was present on this island. And we're not sure yet. We're still investigating and seeing how it's spreading. Did it come from Mozambique to Mayotte and then spread to the Comores? That's something that we can't answer currently, but we are working on that. So in the Comores, after we first identified it, we went with some colleagues from the FAO. They've been supporting our actions quite a lot. We went to visit and kind of assess the situation there in the Comores. And there's currently a TCP, a FAO TCP approval in progress, but we've done some first surveillances on all 3 of the islands, and sample analysis is currently in progress so that we can see how far this is, how far that's actually spread, and what type of containment measures we can implement. So initially, like I told you, the disease was first reported in 2013 here in Mozambique on 2 commercial production sites. So we know that it's probable that farmers with their vehicles visited this farm and then spread it to the other farm. But other than that, there hasn't really been, we haven't found TR4 in the neighboring countries, which is quite surprising to us, of Mozambique and Tanzania, where there's a lot of banana that's grown. Now, we do believe that the actions that we employed on the farm played a role, and that it was found on a commercial farm property, because when it's in small-scale production systems, it's very hard to deal with this disease. So you can see here the type of things that we implemented. All visitors have to park their cars on the outside. They have to go through sanitation, take off their shoes. The farm is providing some boots to them, so that no outside people can come in, and now inside, anything that's inside is staying inside. So these are the type of protocols that we put in place. We also did regular surveillance, which I will talk to you about a bit later. And we've done a lot of awareness and capacity building in the region. But we do believe that it's also something to do with the banana production that's restricted in northern Mozambique. So other than these 2 production sites of commercial production of banana, there's only like these kind of small pockets of cooking bananas. It's a blago type. And it's very dry in this region. So if people were to go onto the farm and take some of the Cavendish material, it would not necessarily survive, because Cavendish needs to be irrigated in this region because it's so dry. So we think that all of these things played a role why the disease has not spread more widely on the African continent. But of course, we have to keep on looking for it, and that is what we are doing currently. So after the introduction into Mozambique, we worked a lot with the FAO on a project called the STOSAR project, and one of the actions that they tasked us was to look at the risk for all of the SADC countries. We use these type of factors to infer this risk. We look at proximity and interaction with TR4 affected countries, what type of banana varieties are grown, because we know that certain ones are more susceptible, how porous the borders are, the capacity and resources to diagnose and intercept plant material, how important banana is in each country, the biosecurity measures that are in place, and then is there large-scale and small-scale commercial production of Cavendish, because there's always a risk when there's these like commercial production companies that move international staff all over. They always pose a really big risk for to spread the disease. And then, of course, also the movement of people, vehicles, fruit, and planting material from these infected sites in Mozambique to other countries. And we came up with this kind of risk map here, where we tasked some high-risk countries. You know what? It doesn't want to move on. And okay, but let me just go back. Okay, so one of our challenges is, of course, this. I think this is a kind of universal problem in many African countries, that the borders are quite porous and there's really a lack of biosecurity. As you can see, this is the border between Malawi and northern Mozambique. People are just walking over that border. And then the other problem is like the widespread sharing of planting material. So banana, it's very common for people to just take suckers and move this material. They will move it within the region, but can also move it to family that's on the other side of the border. So this really severely makes us very, very vulnerable to the disease. we mapped kind of like the banana that's produced in Africa, came up with this map. I have to make a disclaimer that the statistics that we use to agree FAO stats and that came from government statistics, and we're still in the process of kind of mapping more accurately where bananas are grown, because this gives us an important, this shows us where we should focus our actions, where susceptible varieties are grown. But you can see a lot in Southern Africa. There's a highly susceptible Cavendish that's grown in blue. Then there's the East African highlands and plantains that you know that's grown in West Africa. And then there is the diploid East African highland bananas, the Masharis or Muraru bananas that's grown in Kenya and Tanzania, and also some in Madagascar. And these, with some of our initial screenings, they look like they can also be susceptible. We also use this type of risk management approach where we looked at what are the legislation and regulations, what is the capacities to do early detection and containment, as well as research and extension. This we just did for the SADC region, but of course we want to expand that to all other regions, East Africa, West Africa, North Africa, or ultimately for the whole continent. And we kind of made these high risk countries where, as I've shown you, the previous factors that we considered. And we initially, when we did this, the disease was still not actually reported in the Comores, but that just goes to show that it doesn't always help to, you can use these factors, but other things that play that you're not always thinking about. But at least we made this for the SADC region. We also developed this multi-component contingency plan for TR4 in Africa, and I'll kind of discuss each of the pillars of this contingency plan. So the first one we focus on is policy and regulation. The second, awareness. Third, biosecurity, clean planting material, capacity to deal with TR4, surveillance, and research and development. And I will be discussing each of these pillars of the contingency plan in more details. So we've done a lot on policy and regulation. So during the STOSAR project, as I've told you, we've developed a regional strategy to deal with banana fusarium wilt in Southern Africa. And we really want to, it would be ideal if we also have something similar for the West African region. During this project, we also developed a policy brief. It is available online for you to check out on the SADC, and you can look into the STOSAR project to download this policy brief. And we've done a lot of recently workshops on contingency plans. So last year we did two, one in the Southern African region where Lova and George Mahuka from IITA was also involved, where we combined BBTV and fusarium wilt. And we included all of the SADC countries in this training. We've also done Kenya and the East African region. And most recently, we did run a contingency planning workshop for the West African countries in Togo. Part of our actions was also when we first introduced, or when TR4 was first introduced into Africa, we established the African TR4 Consortium, and we would be glad to have more actors in the consortium. We are also regarded as the center of excellence, so we can do the first identifications and we can help developing contingency plans or actions on how to do containment, etc. Our key strategic activities include the monitoring of TR4, how to enforce quarantine regulations, capacity building, research and development, awareness, collaboration between all of our diverse stakeholders, policy interventions, and then the establishment of the TR4 Center of Excellence. We've done a lot on awareness, and we are happy to help with this in your country. So here is some examples of some awareness material we developed when TR4 was detected in Mozambique, and you will see it is in local languages. We're very happy to do that. We've also had a lot of consultations with farmers and with we've had a lot of consultation with farmers and government officials to make everyone more aware of the disease. Biosecurity is a big challenge for us on the African continent, especially because of the nature of our banana production systems. But there is things that we can use, especially when we have commercial production systems. I think in Nigeria, there's also quite a lot of commercial production of bananas as something that you could look at to protect your country. There's different levels of biosecurity, your pre-border activities, on-border activities, farm-border activities, and on-farm activities. I've shown you this picture, but that's what we kind of employed in Mozambique. But this was in Ecuador, and we've also been involved in when TR4 was detected in Australia. They did a lot. I think they're the best at biosecurity in the world. But there is these kind of manuals that we can use as a best practice. There's many pre-border activities that I think we can look at, obtain knowledge on FOC, and this is what today is all about. Assess your country's ability to prevent and respond to incursions of FOC, raise awareness, standardized materials, national capacity. Shakiro just visited us earlier in the year, and he was one of our trainees. So that's very important, and for him to extend this knowledge to other officials, scientists, extension officers, etc. The brochures that I've just shown you is very important to do awareness raising activities. And to introduce an emergency fund. That's something that's very difficult, but it's important. And then to develop an entrance risk analysis and identifying high-risk entry points. Biosecurity, like I said, for small-scale farmers are very difficult because mostly the planting materials are suckers or bits, so you don't know if this material is clean. Sanitation is mostly neglected, and small-scale farmers really cannot contain TR4 by isolating infected plants because they only have such a small plot to work with. But good things is that they are growing banana and mixed cropping systems, and that producers are more flexible to plant-resistant varieties. So if you're going to a commercial company, they only want to grow Cavendish, but small-scale farmers are more open to growing different varieties. They can apply the following measures to protect their farms from the introduction, awareness. You could do micropropagation operations that provide clean planting material, control the movement of people and animals, which is very difficult, but then at least know and learn to identify and report suspected plants. So within this context, we currently have a project with the Australian government, the Australian Centre for International Agricultural Research, where we're looking at these small-scale production systems in Mozambique and Tanzania. And we try to see how can we optimize biosecurity, or how can we implement it in such small holder farmer systems. Clean planting material I've talked about a lot. There's some countries that do have macro-propagation operations as well as tissue culture, but we know that on the continent, it's still something that's very expensive for farmers. So it's not very well practiced. So we really need to work on the clean planting systems on the continent. That's one of the things that we really need to look at. Then we've done a lot of capacity building over the last 15 years, I would say. We do a lot of trainings and awareness, such as today. We're sharing our knowledge and making people more aware. Shakira just visited us on a very intense training course where we did prevention and management of the disease. And so we are happy to help with this type of actions as the Center of Excellence. We've done a lot of surveillance, particularly in the Southern African region. In that the STOSAR project, we've received over 600 isolates from different Southern African countries. Most of the time all of the time we only found race 1. We didn't find TR4 other than in Mozambique and Mayotte Island, and of course the new island of Comores. But we constantly need surveillance to be able to track the spread of the pathogen. We can't just do surveillance once because the pathogen can keep on spreading. And we know we need to do more surveillance in Eastern and Western Africa. I think we're working a lot with our colleagues there after our training and the contingency workshops that we recently did in Togo. We really want to do surveillance so that we can really make sure that TR4 is still not in any of your countries. We've also developed with the FAO a digital surveillance tool that can be adapted for surveillance in your country. So please contact us if you're interested to do surveillance and we can support those actions. Lastly, I will just talk a bit about resistance. Developing resistant bananas is really the pillar of management of this disease, because once it's introduced, there isn't much you can do about it, but breeding is very difficult. And I think Lava will probably touch on that as well. Conventional breeding takes a long time, and a lot of the time, the end products is not acceptable to consumers. There has been some breeding, some resistant varieties, the FIA hybrids that are planted quite widely in Africa. But I think still we could say that a lot of the time the consumer don't accept those varieties. So we can look into kind of biotechnology, genetic engineering, but the problem is that GMOs still have, especially in Africa, quite a negative public perception. There is a GMO banana that's resistant to TR4 that's been released in Australia, but GMOs are still not accepted in most African countries. And then we can also look to mutation breeding, either through somoclonal variation or chemical or physical mutagenesis. There has been a somoclonal variant that has been quite successful in the Philippines. These varieties are not completely resistant, so they need to be supported by good agricultural management practices. But yeah, they are currently the only option for the commercial production systems when TR4 is introduced. And we've also looked at these varieties in Mozambique, which I will talk about a bit later. So here is some work that has been done in Australia on kind of popular varieties. And you can see that FIA varieties most of them are doing quite well. These are the somoclonal variants that you can see. They do get disease, but at lower levels than the control. And then some other ones that are also resistant. But none of these are going to be is ready to replace the Cavendish as a commercial variety. Before TR4 was even introduced into Africa, we took a few varieties and we kind of preempted its introduction, and then we tested these varieties in China. And you could see that most of the East African highland bananas, they were not immune, but they developed much lower disease, as well as some plantains, but we've only tested limited of these East African highland and plantain varieties. So we really want to do much more on the continent, the same with plantain varieties in West Africa. Then lastly, I would just like to say that we're also looking at sustainable banana production systems, especially in small grower systems. We try to see what is the effect when you're intercropping. This will not eradicate the disease, but it might make you more resilient to when the disease is introduced. So there's many type of things that have been used in banana production systems, including intercropping, crop rotation, cover crops, and as well as bioorganic fertilizers and such that can be considered when TR4 is introduced to make banana systems a bit more resilient. So to end off, the key points that I want to leave you today is that we are extremely vulnerable to TR4. Cavendish production is very important and it's wide, it's produced all over and it's highly susceptible. We are mostly using suckers, not tissue culture material, which severely exposes us, and then the porousness of our country borders. Biosecurity measures, awareness campaigns, and the availability of TR4 resistant bananas are fundamental to deal with the disease. We need to do regular surveillance to assess the spread of TR4 in Africa, and all suspect cases must be reported immediately. All of our efforts should be made to contain TR4 at the contaminated properties and protect small-scale production, because when it's introduced into these systems, it's going to be really, really hard to deal with it. Lastly, we need to focus on political will, commitment, and proper investment in plant biosecurity and phytosanitary measures that is required to deal with TR4 in Africa. Thank you so much for the opportunity, and I'm happy to take any questions. I think it will only be after the other two speakers.
Yeah, thank you very much, Musat. Thank you so much. That was just hitting the nail on its head. Thank you very much. As you have rightly put it, please, you may want to compile your questions, or if you want, you can post them on the chat box, so that as soon as we take the other two presentations, our distinguished guests will begin to respond each to the questions raised or contributions you intend to make. So at this point, I will call on Dr. Kazim for the next citation of the other speakers. Over to you, Dr. Kazim.
Okay, thank you, Dr. Chris. When we take the question, please just put it like the chat, and then later on you can raise up your hand if you want to talk physically. Going forward, also we also be taking the messages that we left behind when we finish the two presentations. So we want to do the citation for Dr. Lavakuma. Why I share is sorry. Okay. So, Dr. Labakuma is the leader of the Plant Health Program and the head of Biology, Molecular Diagnostics and Germplasm Health Unit at the International Institute of Tropical Agriculture, IIT in Ibadan, Nigeria. Dr. Kuma is a biologist. He earned his PhD in biology in 2000 from SV University in India, with a remarkable career spanning over 25 years. 17 of which were dedicated to plant health research in Africa. His program involves the characterization of viruses, the development of diagnostics tools, and finding solutions to control the spread of virus disease in important food and horticultural crops in sub-Saharan Africa. He initiated work on the banana bushmeat virus, BBTV, in 2008 to map the continental spread of BBTV. He was involved in the first discovery and official diagnostic confirmation of BPTV in 7 African countries, including Nigeria. In 2012, he initiated Alliance for the Control of Banana Bushmeat Disease in Africa for coordinated management of an important transboundary virus disease ravaging bananas in the continent. As part of the CGIAR Germplasm Health Program, his program also deals with the prevention and control of several transboundary pests and diseases, including maize leather necrosis and cassava brown streak disease. His program developed novel tools for virus disease surveillance, including remote sensing methods, and organized several capacity development activities. and group trainings in virus diagnosis and disease control. His program also developed procedures to produce virus-free planting material. He has authored and co-authored over 140 research articles in peer-reviewed journals and guided several MSc and PhD students. He is a recipient of several merit awards and several international. He has served on several international panels, including the steering committee of the African Union, Inter-African Phytosanitary Council, as well as the IPPC African Phytosanitary Program and the Executive Committee of the International Plant Virus Epidemiology Committee. Today, he will be giving an update on BBT status in Africa with an emphasis on the virus disease status in Nigeria. Thank you, Dr. Lawakuma. You have the floor. You can share your screen, sir.
Yeah, thank you, Kazim. And I would like to thank organizers for giving this opportunity. And it's a pleasure to give a lecture after Dr. Diane, who has done a fantastic job in introducing the Physiarum wilt challenge in Africa and why it remains a problem and why it is such a big challenge to control because of the way the crop is cultivated. Many of the control measures that Dr. Diane presented in her talk are relevant for bunch top as well. So it's very important. I hope we can see my screen.
Yes, we can see your screen.
Okay. Are you seeing the full screen or the presentation side of it? Can you confirm? Are you seeing the full screen or the presentation side?
We are seeing the full screen.
Okay, good. So in this talk, I talk about another important threat to banana, which is a virus. And unlike Physiarum wilt, this virus has been known to occur in Africa for more than 100 years at a continental level. First, very briefly about the crop. We all know banana in the continent, the production. In many countries around the world, banana is a desert. But in many of the developing countries, especially in the global south, it is a very important food security crop. And its uses is tremendously diverse. It is cultivated for many purposes, from food to processing, and also for industrial uses, textiles, paper. There's a tremendous applications, and it is highly commercialized crop in many countries. An interesting fact is that it is also one of the most subsistence crop. For instance, it is grown with very limited inputs. In the world, according to the data, excuse me, data available in FAO, more than 50% of the banana production area is in Africa. But then if you look at the contribution of the production, it's very low. It's only about 35% of the total production. There is an error in the figure here. About 35% of the total production, global production, is in Africa, although the area is more than 50%. That implies that the yields in Africa are very low. Okay. But the cultivar diversity is very high in Africa. There are different types of varieties grown, the plantain, banana for cooking types, desert types. So there's a huge diversity. And the very important clone that is very widely grown in East and Central Africa is the highland bananas. It is considered as one of the largest land masses of a single species, single clone cultivated in any part of the world. The diversity is staggering. Like if anybody working in banana, they see tremendous variation. It is one crop that you would encounter in almost in every location. Even as the driest part, you can find banana in a backyard. So most often it is grown as a mixed crop. Intercrops are also very common, especially along with cocoa. And then monocrops are limited, but they are growing. It's a growing trend, the monocrop cultivation of banana. And this is mainly for industrial purpose or export or completely commercial focus. So with many big companies coming into Africa, they are focusing on monocrop cultivation. And also many progressive farmers, the youth farmers that governments are encouraging, they're also going into monocrop banana production with drip irrigation. And then there is no need to say about the importance of it. And as a backyard crop, almost every backyard have a banana. And also orphan plants. This is one thing that you will see that there are bananas which are not claimed as anybody's crop, but it is growing along the roadside, in the forest margins, extensively. It's almost because the banana has a very interesting architecture. It can survive extreme weathers, and then the underground stem will continue to produce new plants. So these orphan plants are widespread, and sometimes people harvest the fruits, but the plant nobody takes care. They're just there. And it is a crop that is grown in rural areas as well as in urban areas, and also in peri-urban areas. In deep forest, you can go inside the deep forest. There are banana plantations. Why I'm mentioning all of this? It's very important to understand the cropping system. If you want to deal with the disease management, you need to understand how the crop is grown. So we are dealing with a situation in Africa where we have completely different aspect. Is banana is almost grown as a perennial. Okay, it's not an annual crop. It can be grown as an annual crop, like farmers can harvest fruits and then they completely replant the whole field, which happens in many monocrop situations, especially in advanced farmings. But in traditional farming, it is almost grown as a perennial crop. Once established, it will grow continuously. So that means if any plants get infected with any disease, then it is going to survive forever. In terms of, if you look at the banana, it's a very important crop in Nigeria as well. According to the production data, there is an increase in production area in the last 20 years. There is also a change in the yields. It used to be at an average around 6 to 7 tons per hectare. In the last 4 years, Nigeria is registering 4.8. Sometimes these data could also be the way the data is collected by FAO, but in general, the growth of banana is positive. Now coming to the bunchy top. Having said that, it's a very important crop, especially in Africa. It's the fourth important food crop in Africa. There are diseases that are really threatening the production. We heard about the Fusarium wilt. The other threat is banana bunchy top virus. It is a virus. The disease name is banana bunchy top disease. Most lethal virus to banana, and it completely destroys the production. The images that you are seeing these are not suckers or young banana plants, but these are the bananas that were affected by the virus. This disease derived the name bunchy top because once the plant get infected, every new shoot that emerge from the infected source automatically remains stunted. So it appears like a kind of a young sucker, but in fact it is a effect of virus. So this is the disease that was first reported in Fiji in South Pacific region in 1889. At the time, it was just recorded as a disease because nobody knows the cause of it. It was suspected as a virus disease much later based on the transmission properties, the symptoms, but the virus itself was not identified until 1990s. It was known to be one of the smallest virus particle. It has just about 20 nanometers. And because of the impact on the crop, it is classified as it is one of the most important threats to banana biodiversity, and included among the top 100 invasive species in the world. This invasive species list includes animals, plants, everything. So for a virus to be named among the top 10, you can understand how significant it is. And in most countries classify bunchy top as a A1 quarantine threat. Okay, it has accorded a highest threat level. It is transmitted by banana aphid, Pantalonia nigronervosa, and also through infected planting material. The virus basically has a single-stranded DNA as its genome. Each single-strand DNA encodes one protein. I would not go into details of it. But what is most important is each of these DNA component is encapsulated by separate particles. When if it transmits the virus, it has to acquire all the 6 segments. Otherwise, the disease would not establish. So in terms of, it's a very complex pathogen. Each of these virus multiplies independently as long as there is a replicase protein. But all 6 have to be transmitted together by the aphid for infection to establish. All the bananas, regardless of which variety is grown in which part of the world, almost all the bananas that are grown today in every part of the world are susceptible. There is no resistance to bunchy top. There are some diploids that have been known to be resistant to bunchy top, but these diploids are not edible. So currently there are efforts ongoing to see how this resistance can be transmitted to cultivated varieties. Apart from banana, the virus is also known to infect some other host species, such as the NZ and a few species in gingerbread ales. But in terms of epidemiology, these other hosts are likely to be dead-end hosts. Banana remains the most important host for virus, as well as the vector, and it remains the major source for virus transmission. That doesn't mean other hosts are not significant, other alternate hosts are not significant, but they're very less likely to be a threat in terms of posing a risk for further spread. It's very important to understand that this banana aphid, Pantalonia, is highly specialized and it prefers to feed almost exclusively on banana. It doesn't survive. If you transmit this aphid to other hosts, it won't establish, it won't colonize. So it has a kind of very unique association. Another important fact is this aphid is present everywhere where the banana is grown. So that means this banana aphid is present everywhere. If by chance the virus get introduced, there is a host, there is an aphid, automatically the virus can spread further by this vector. So you can see in areas where there is no virus, but you can see the banana aphid. And these are very tiny and most often they are present on the pseudostem, not on the leaves. Finding this aphid on the leaf is very rare. Since pseudostems are always covered, unless you peel the pseudostem, it is not easy to see this aphid. So they are not readily visible unless we specifically look for it. Most often they are wingless forms. They only develop wings when they identify some threat or when the plant is under stress. Otherwise, they continuously feed on the plant they colonize. This aphid transmits the virus in a persistent form. Apart from aphid, the other major mode of transmission is the planting material. Banana is a vegetatively propagated crop. When you harvest any infected tissue from infected plant, and when it is used for propagation, automatically the virus multiplies. So be it a tissue culture, be it COM or macro propagation or suckers, as long as they are harvested from infected sources, the virus will multiply. And this virus do not kill the plant. Even tissue culture plant, the virus do not kill. So that means if there is a virus in micropropagated plant, it survive, and when it's introduced into the field, the virus spread. So in terms of global distribution, it is present in many countries, almost 36 countries around the world. Mainly, most of the countries in East Asia and South Asia are affected, and then Africa. In Africa, there are 16 countries where this virus is present. In sub-Saharan Africa, it is present in about 16 countries. The banana aphid, as I mentioned, is present in all the countries. There are two different lineages. Technically, it is only just one species, but there are some genetic differences that can be distinguished to identify isolates in Southeast Asia. There is only one haplotype that has spread to rest of the world. All those areas indicated in yellow, this is the same isolate present in Africa. As you can see, how this kind of distribution basically implies that the virus is multiplying through the planting material. So this is the typical situation that you observe in the field. This is the typical banana bunch top infected plant. You can see there are some fruits emerging from some pseudostems, which are the shoots that have emerged before the virus infection. Every shoot that comes after virus infection, they are stunted and they do not produce fruit. And in a late infection, you can see a kind of deformed fruits, which are small and unmarketable, not edible. But in most cases, there is no fruit production. Okay, all these plants that you see, these are not suckers. These are all the virus ravaged bananas. The loss is 100%, and this happens within one season. And it is often called as green plague because the plants remain green, unlike Physarum wilt, as you have seen in the photos. The Physarum wilt infected plants turn yellowish and brown. In case of banana bunch top infected plants, they remain green, and farmers don't see this as they have infection in the farm. So this is a big problem. Even if there is infection in their fields, it takes very, very long time before they realize that they have a problem in the field because the plant remains healthy. I mean, they look apparently healthy, and then farmers think there is just slow growth because of the soil or some other problem. And this is a picture recently taken in Tanzania. So this farmer field was affected. You can see this is pretty much the last harvest that farmer is going to get from his farm, and there is no further production from this field. And he, in fact, have harvested some of the infected suckers, and these are all the new plants that he established. He doesn't know that he is actually planting the infected sources. And this is a classical example. This is the field. The farmer has weeded, and he's still hoping that these plants are going to grow, and he's even putting fertilizer, assuming that this stunting is because of some soil issue. And then once they realize that it is no longer productive, they just abandon the crop. After they abandon, what happens? The weeds will grow. You can still see the banana, which is infected with bunch top, still surviving. Such surviving plants serve as inoculum sources for aphid-mediated further spread. So this is a very difficult problem to manage. And this is again a new farm established. The farmer has bought suckers from infected plant, and you can see these are infected, but he doesn't know that he actually harvested infected suckers and established a new field. So this is the loss is tremendous. As you can see, this is a picture taken in Benin in 2011. The farmer was assuming that when we talked with the farmer, he says she's expecting fruit from it. This is what the farmer is expecting. The reality is this is the bunch top. There is no more fruit coming. So in a disease affected area, before disease, they used to harvest a truckload of fruits. After bunch top, within one season, all they could harvest is just enough to send on cycle. So there are no accurate estimates of production losses, but based on the modeling and the limited impact assessment studies have identified that bunch top annually causes yield losses worth of more than 200 to 600 million per annum. This is a tremendous loss. So this is one of the high threat recognized in Africa as one of the most invasive alien species, causing tremendous damage to banana production. This was the situation in Africa. The virus was, as I mentioned, it was known for more than 100 years. The first report came in Egypt in 1920s. And then the next report came from Central Africa in DRC, which was in 1960s. It may have been there before, but the first report was made in 1960s. And this spread from DRC, the virus has spread widely in many sub-Saharan African countries. As of today, we know that the virus has spread to 17 countries, and including Egypt in North Africa, total 18 countries are affected by banana bunchy top. All these have been officially confirmed. 7 of those have been coming from since our surveys started in 2008. So this was the kind of the situation when we started work. The virus was known to present in Central Africa and Malawi, and in North Africa and Egypt. And then when we started surveillance surveys, we could able to map the extent of spread in each of these countries and also identify new spread in Cameroon and Angola. And then as we expanded our surveys, to our surprise, we found it in West Africa, right at the border between Benin and Tanzania. This was in 2011. First detection happened in Benin. After finding the virus there, we suspected the virus may also be present in Nigeria, and we expanded the surveys, and then we have realized the virus is also present there. So at the same time, there was reports of virus in Mozambique and Zambia and South Africa. The further confirmations came soon. I just focus a little bit on in Nigeria. In Nigeria, the disease was detected in Ogun State, very close to Benin border. And as soon as we detected, we immediately worked along with our quarantine agencies. As you can recognize some of your former colleagues here for first confirmation, and immediately we started campaigns on mitigation. There are 3 local government areas affected by bunchy top: Uva North, Uva South, and Ipokia in Ogun State. We also have done surveys in other parts of the country, and fortunately, there is no evidence of virus spread, including the most recent survey was done this year. And we continue to, it is not possible to visit every field, but we continue to crowdsource for any risk or any apparent reports of bunchy top, but there is no detection outside this area. this is actually a fantastic result, considering that the disease is present for more than 11 years known for more than 11 years in Nigeria. And we managed to contain, thanks to Nigerian Agriculture Quarantine Service for effective monitoring and preventing inward spread of planting material. There are also other reasons, epidemiological reasons, why the virus is limited only to this region. And there are a number of other geological boundaries, as well as the way the planting material is moved. So this area is basically sink. Nigeria don't source planting material from this part of the region. So that is also a kind of a blessing. So today, this is the situation. There are many countries where the virus has become endemic, which are indicated in orange. In other countries where the disease is present, it is localized. It has not spread every part. Okay, so that means there is still a scope for containment or eradication. We also have done some studies to understand how the virus is spreading, and we confirm that there is two different introductions into Africa. And Egypt spread is different from that of spread happening in sub-Saharan Africa. So it is the virus that was detected in DRC. It is the same source spreading into other regions. What does it imply? It means the virus is either spread by the aphid or through the planting material. It is the same source. Okay. So basically, there are two types of spread happening. This is the most recent spread, East Africa. As you can see, there is a disease here, but there is plenty of areas there is no disease. What does it imply? It means somebody have moved planting material from one region to another. So this is what we call jump spread. So jump spread is very dangerous in terms that it contributes to long distance spread of the virus. Okay. And then contiguous spread, that is from farm to farm. It happens because either people are exchanging planting material within the community, or the aphids are spreading the disease. So this is what is happening. When we try to understand what is contributing to the spread, it is mainly because of the jump spread and contiguous spread. Long distance movement is only happening because of sucker taking from one area to another. In such situation, mostly men are doing that compared to the women. And also, once it is introduced, if it is there, and then it is contributing to the spread. So these are very classical. Like, we have taken a lot of pictures when we are doing our surveys where people are literally moving infected material. They don't know that they're moving infected material. We need to be very careful when we attribute this information. Farmers don't have information. They don't have awareness. So therefore, they think the infected plants as suckers, healthy suckers. They're harvesting and moving. So awareness raising is going to be very, very important. So this is a situation here. As you can see, in many farms, farmers simply abandoned because they could not manage. They abandoned. They changed to different crops. But since the virus do not kill the plant, this vestige always survive, and these are danger because they serve as inoculum for aphid-mediated spread. So how do we manage such a complex problem? It requires integrated method. There is not a single method that we could use. It requires surveillance. It requires quarantine containment. We are also focusing on trying to identify host plant resistance. I will just talk about it a bit later. Cultural control, biocontrol to target aphid vector, and then understand the epidemiology and ecology, which is very important. If we understand the disease, how it is spreading, then we can come with these control measures. So we have come up with a strategy in 2013, bringing a kind of a sequential how we manage it. It's called, we created an alliance because it's a transboundary disease present in many countries. If one country take control measure and if neighboring country do not act, it is of no use because the virus will still spread from the neighboring country. So it requires a coordinated approach. So we brought together many countries which have BBTT and established a kind of a standardized framework which countries can adopt. So these are the countries where we are currently focusing. We could have worked on main countries, but funding is a limitation. Most recently, we also started working in East Africa following the discovery of the virus spread, Tanzania, Uganda. The virus is not present in Kenya, but since it is close to Uganda and Tanzania where the disease is present, they also have taken some proactive measures in terms of active surveillance, raising awareness, etc. So what tools are available? This is important, okay? So generally, for any disease management, we need three important things. One, surveillance, scouting. We need to go and look for disease. And number two, we need diagnostic tools for confirmation. And number three, how do we eradicate or control the disease on the farm? So we have a range of diagnostic methods from ELISA to PCR to real-time PCR. But the most useful technique, especially for quarantine monitoring and serial certification, is the rapid test, which doesn't require many expensive laboratory setup, and they're very easy. We can get results in 15 minutes, under 15 minutes, and they're also, each test costs about $2. So this has been now perfected. This method can be utilized for testing virus in both plant as well as aphid. Many of you have experienced some of these methods during the COVID time. This example here is called recombinant polymerase amplification. It can be done in a very limited lab, limitedly equipped labs. It can also be used under field setup. And we also standardized a simple testing which doesn't require any. So if you want to do a rapid test in the field, we can just directly take leaf tissue, and then we have simple workflow. Within 5 minutes, we can complete that workflow. In minutes, the reaction will complete, and we can see the results. And we have been organizing training workshops to scale these methodologies. In addition to that, we also have developed a mobile app that would help real-time reporting, and also it creates maps so we can actually spot where the disease is, and we can confirm. So these kind of tools will actually help coordinate the surveillance efforts and also real-time approach. Surveys are expensive. We are also now looking at the remote sensing approaches, how we can actually detect virus using remote sensing data. Surveys, for example, it takes we can only do 10 communities if you have a good road and vehicle. But with remote sensing, we can actually cover 300 hectares in one day. All you requires is a good program. Currently, we are designing very machine learning methods and artificial intelligence tools to identify bananas, and then combine that with surveillance data to risk model and then predict where the disease is present. So using these approaches, we can actually economize the surveys, and then we can target, instead of going and looking for disease everywhere, we can go and targeted surveillance, so which reduces the cost. I cannot emphasize the importance of the awareness creation. It's very, very important. Unless people know what is disease, it is not possible to contain this problem. Since it is a difficult disease to recognize because of a lack of very typical symptoms, such as browning or yellowing, we need to raise awareness. And this is where the community engagement is very important. We have been using all levels of engagement. Again, in Nigeria, for instance, Nigeria Agriculture Quarantine is doing a great job. And also NIHOT, we have been working with NIHOT and other stakeholders, including policymakers, politicians, because they are influencers. They have a voice. They can, when they speak, people listen. And so it creates higher impact. So we have been doing this periodically in the areas where the disease is present and also where there is no disease. In terms of management, we don't have many options. The only way is to kill the infected plant by injecting with herbicide, because the virus do not kill the plant. We have to eliminate. Otherwise, the virus sources persist in the field, which is a threat. And we should also eliminate aphid vectors, especially on the infected plants. And we generally do that by using soap water, not insecticide, because in most often these are present in the communities. There are small ruminants. If you spray insecticide, if the ruminants go and eat them, and sometimes they cause a problem to the small ruminants. So we advise soap spray. And if there is no ruminant risk, then insecticides can be sprayed. So these are the only two options if you actually want to eradicate infected plants. We have been looking at the host resistance by screening the germplasm that we have. We identified some genotypes which have better tolerance. They are not immune for both aphid and vector for the virus, but it is still a long way. We don't have any varieties to recommend for farmer cultivation so far. We also have simultaneously ongoing programs on breeding and also transgenic and gene editing methods, but these are all long-term solutions. For time being, this is the only option we have for disease management. Remember this timeline. If you want to manage the disease, it takes 3 years. Patience is very important. Why? We had to first start with the eradication of infected mats. We had to source clean planting material and then replacement with clean planting material, eradicated fields. And we need to continuously monitor because there is inoculum, there will be reinfection. So it's very important to follow the sequence. If we abandon these steps, the infection will reappear in the field and all the efforts that have put in place are not going to be effective. So patience and a persistent approach is critical if you want to manage the disease in areas where the virus is already present. It's a long-term game. So lastly, contingency plans. As Dr. Diane mentioned, we have been working with a number of stakeholders, more recently with FA1 Inter-African Phytosanitary Council, along with quarantine agencies. We have been systematically working with a number of countries to raise awareness as well as develop national plans with regard to how to manage. So these plans consider two aspects: the countries with disease and or the areas within country with the disease and no disease, because the management tactics are different. If there is a disease and if there is no disease, then it is mainly about exclusion and prevention. So in both cases, awareness raising is very, very important. We have been developing joint contingency plans for both BBTV and TR4 because many aspects of the contingency plans are applicable equally for TR4 and BBTV. So in this regard, we have been now combining our forces so that we can be more effective and efficient when we message and work with our quarantine partners. The most recent workshop was held in Togo for Western Central African countries. And before that, we have organized one in Southern Africa. There was also an effort in East Africa done in May last year under the coordination of Inter-African Phytosanitary Council. So another aspect which we are also planning with the Nigerian Agricultural Quarantine Service is a surveillance system for Nigeria for tracking emerging and invasive risks, a real-time reporting system using both expert as well as crowdsourcing approach. This is just a concept diagram. And we have proposed this a while ago for funding, but unfortunately we were not successful. But now again, we have reinitiated this plan and hopefully we will set this up because now we have more threats like TR4, bunchy top, we have maize lethal necrosis, we have cassava brown streak, dead palm weevil. There are many problems. So a tool like this actually will help coordinate surveillance system. So this is my last slide. When I talk about bunchy top, I don't forget that there are other threats. There are threats such as bacterial wilt, Fusarium TR4, which have limited distribution in the continent, and understanding where they're present, understanding the symptoms of this disease and their epidemiology, how they spread, and what measures need to be taken to contain them is critical. So we often advocate countries that it is very important when we think of contingency plans, all these three threats need to be considered. So with that, I thank once again organizer for giving me this opportunity, and we have a number of partners contributing to this work, and I take this opportunity to thank each and every one of them.
Okay, thank you, Dr. Laba, for a very good presentation. We have been enlightening more on the banana bushy top again, and then we will take a further question. Please, you can put your question in the chat or raise up your hand when it comes to it. But before then, let us take the last presentation, and that would be by Dr. Bume Bitoye. So I'll be reading a citation. Dr. Dorcas Olubunmi Bitoye is a plant breeder and assistant director research at the National Horticultural Research Institute, NIOT, with a decade and a half of vast experience in plant breeding and molecular breeding design, pre-breeding of germplasm resources for efficient conservation and utilization, excellent participatory on-farm iterative and demand-leading plant breeding skill. She possesses excellent skill in statistical analysis and interpretation using SAS, SPSS, and aerostatistical software for phenotypic, genotypic, and socioeconomic data. She started her educational journey at the prestigious and oldest guest-only institution in West Africa, St. Anne's School, Molete, Ibadan. She obtained her first degree in botany from the University of Ilorin in 1998, master's degree in plant genetics and breeding in 2003 from the University of Ibadan, and her PhD in plant breeding in 2015 from the University of Ghana. She was selected as a fellow of the University of California African Plant Breeding Academic Class 3 in 2018. Dr. Obi Toye is a recipient of several awards due to her hard work and diligence, among which are the Norfolk OKP Scholarship in 2021, the beneficial and focal person for the C224D2 I1 project 2020-2023. She's a fellow of African Plant Breeding Academy. She has received travel grant award. And then she has several grants as a co-principal and investigator, rising up to some $41.7 million dollars. She was recognized as the best female president in the Faculty of Science at the University of Ilorin, Kwara State in 1996-97 sets, and she has also been recognized as a quality leader, as the president of ISAF IIT in 2014 and the most influential scientist in NIOT in 2018. Dr. Ibitoye has attended and presented papers in several training workshops within and outside Africa. She has to accredit 16 published conference proceedings, both in local and international journals. She has 21 articles in peer-reviewed journals and 2 book chapters. It is only natural to expect that one who has gained so much so far will be source for knowledge transfer. Dr. Ibitoye is a part-time lecturer at the Police University of Ibadan. That's Pan-African University. Furthermore, she has supervised 8 MSc students, 4 BSc, all from the University of Ibadan, and currently supervising 3 MS students from the University of Ibadan and the Pan-African University. She is a member of so many professional body like the African Plant Breeders Association, Nigeria Plant Breeders Association, the International Society of Horticultural Science, the Genetic Society of America, Crop Science of America, Crop Wide Relative Specialist Group of the world, and of course, the Horticultural Society of Nigeria. He's actively involved in a program for seed system innovation for vegetative propagated crops in Africa, that's PROSIVA, which aim to improve the efficiency, productivity, and profitability of vegetative propagated crop seed setup and quality seed delivery to farmers in Africa. She'll be speaking on the certified clean banana-plantain planting material. Dr. Ibitoye Mazim in life is live and let others live. She's joyfully married and the marriage is blessed with 2 adorable daughters. Dr. Ibitoye, I'll be sharing her screen while she does her presentation. Dr. Ibitoye, you have the floor. Can you hear me, Dr. Ibitoye?
Yes, I can hear you. Good afternoon.
Good afternoon, ma'am. Thank you, ma'am.
Yeah, sorry. Thank you very much. Thank you for the privilege given to me this afternoon to be able to speak with this body of knowledge. Thank you very much. My task here is very simple. I am to talk about certified and clean planting material for our banana and plantain. Please, next slide. Can you hear me?
Yeah, we can hear you.
All right. I wouldn't want to repeat so many things because my senior colleague, Dr. Lavakuma, has done a good justice to what banana and plantain is to Africa, West Africa, and Africa as a continent and a different region of Africa. He has talked about the different uses of the crop, as the crop is is an important food crop and even a cash crop in our system, as it can be used for desserts as fresh fruits and also be processed into different products. In this context of my discussion and my presentation, I will be referring to the Musa group as the banana, plantain inclusive. We all know that plantain is a starchy banana, is also in the banana group, and they are both in the Musa genus. Banana is more or less the dessert, the one that is eaten fresh, while the plantain is the one that is cooked, can be boiled and do with other ones. In the West and Central Africa, the Agbagba type is more common, While in the east and southern type, we have the highland bananas, the matoke, the cherry, and the likes that are also the starchy banana group. Next slide. Yes, please, the production has also been dealt with by Dr. Lavakuma as more production is more into the West and Central Africa. And Nigeria comes after Ghana and Cameroon. Please, the next, press next so that we can have the chart. As this is the FAO data for plantain production. Next slide. In Nigeria, plantain or banana is mainly cultivated in the southern states of the country. Even though, as rightly said in the last presentation, that there are different production ecosystem. We can have them growing in the forest, but it is the banana group, the banana subgroup that grows and can survive even in thick forest. For the plantain subgroup, they cannot survive where there is too much heavy weed. But for the banana, the desert banana, they can grow the cooking banana. They survive very high forest. So it is grown in all the southern states of the country, majorly grown in the southern state of the. That is not to say that it cannot grow in other parts, but major production comes from the southern states of Nigeria. We have different production systems, which has also been discussed in the last presentation, can be grown as an intercrop, as a bush plantain intercrop with cassava, also like the one planted in the forestry species with some forestry crops like Melina, arborea, the teak, and etc. And also we can have it in different convenient points, either at the backyard, in the environment. It can be grown in any place. So it can also be grown as a monoculture practice, where you grow only plantain or even banana as a monocrop. Next slide. Next slide, please.
Yeah, you are on the next slide.
Now, we want to talk about certification as it comes to banana and plantain production in Nigeria. It is good for us to know those people that are involved in the production of this crop. From survey that was carried out in 2020, we identified that youth male are more into plantain production, while the female gender are more into the processing and the marketing of the fruits, either for the plantain as a fresh dessert, or as either the banana as a fresh dessert, or plantain as a harvest for product for different processes. Next slide. So it's good for us to also have information about what is the current status of the banana and plantain seed system in Nigeria. Currently in Nigeria, the current status is that plantain banana seed system is highly informal. There is no regulation, there is no certification. So sellers or aggregators gather materials, the suckers, which we refer to in this presentation as seeds, because seed is any proper good that you use to regenerate a crop. So the suckers are aggregated from existing farmlands, from existing mats of plantain, and that is what they aggregate. They can mix up different varieties if they have a huge demand for suckers. They can mix banana with plantain. They can mix different varieties together. This is because there is no regulation, there is no certification for the crop in Nigeria as we speak. And this lack of regulation or certification makes it easy for spread of pests and diseases. It makes it very convenient, because if you go to any mat of plantain and banana, you can have like 4 or 5 suckers. And if someone has a request for, let's say he has a request for 5,000 suckers and he cannot meet it, he can just call people randomly and the people will just go to the mat and uproot. It means that whatever is existing on that mat, where they are getting the suckers from, also goes with that sucker to wherever they are planting afresh. So it means that if this plantation is already infected with BBTV, like Dr. Lavakuma said, the spread of BBTV was made very convenient through sharing. and movement of planting material from one place to the other. So because of lack of certification, it makes spread of pests and diseases in Nigeria as we speak today, very convenient. Also, the marketing of plantain seeds is carried out by producers or sellers at convenient location. There is no specific place or a specific market in any of these growing regions. In any of these different southern states that grow plantain and banana, they do not have specific markets for maybe the selling of the suckers or selling of the suckers. This also contributes to the proliferation of different aggregators across the growing regions. Everybody's selling anything. Everybody's selling what there is no quality assurance. Nobody is checking whatever anybody is distributing. So we can just move material from the south, from the southeast to the southwest, and we are just easily transporting whatever is existing. And at the moment, the suckers of plantain is only being marketed within the country. It is not being exported. So next slide, please. Now we need to know, what do we mean by seed certification? It is a legally approved system for quality control of seed multiplication and production, with a purpose to maintain and make available to the public high quality seeds and propagating materials of known varieties as to ensure genetic identity and purity. I would like us to give attention to the key words in the purpose for seed certification. We want to make the seeds available to the public, to the requesters, to the entrepreneurs, to the farmers. We want to make those suckers, those seeds readily available to them. But we want to make it a high quality seed. If there is no certification, there is no way we can ensure that quality assurance for high quality seeds. We want to be able to know the variety that is being sold or being shared. As plantain is a vegetatively propagated crop, plantain and banana, at that stage of the sucker, it is mostly difficult for anybody to identify whether this is even a banana or a plantain at that very stage where they are already mixed up. Now, that number one is difficult. Number two, it is difficult to say this is the particular variety that I'm being supplied with until those materials start fruiting. That is when you start knowing. So the essence of seed certification or regulation is to be able to have available quantity at the good quality and a known variety, so that we ensure the genetic purity and identity of the varieties being shared. And also, seed certification happens to be the starting point to having a successful crop cycle. And the agency that is saddled with seed certification and regulation in Nigeria is the National Agricultural Seed Council. That is NASC, that is based in Abuja. Now, the work of this seed council can only be made easy with the collaboration of all agencies that is in charge of seed importing and seed exporting, like the quarantine services, like the custom services, and also the NASC. So when they work together and not in cross purposes, then it will be easy for us to have this good certification for quality delivery. Next slide. Now, who are the seed actors? What are the seed classes? Even though we do not have any regulation or certification of plantain and banana like we have for cereals, like for maize, like for soybean, there are seed actors that are across the value chain of every crop. We have the nuclear seeds producer, the breeder seed producer, the foundation seed producer, the certified seed producers. Each of these seed actors is meant to be regulated because each one of them is producing at a level of quality that is expected. For a nuclear seed producer, 100% quality is expected. And so there is no room for any mix-up. There is no room for any mix-up, like it's with at least some allowable percentage can be given to somebody that is producing the QDS, the quality declared seeds, or those ones at the community level. But when it is the nuclear seeds that you are feeding into the breeder seeds, it is highly important that high quality, high genetic purity is expected at like 100%. There is no chance, there is no room given to lesser quality. These are the different actors. And when we are talking about seed certification, there are different people that play different role along this seed value chain. And NASC has the mandate to certify and regulate the production of each of these actors along the value chain so that quality delivery is ensured and there is value for money for the entrepreneurs that are buying the seeds. Next slide. Now, what are the steps towards certification? What should we do? If we say at the moment now, we do not have certification in place, we do not have regulation in place for banana, not for any other, but for the banana that we're talking about. Number one thing, there cannot be a certification for any crop variety that is not officially registered or released in the country. So this has to happen because as we speak now, for banana and plantain, there is no registration, there is no registered variety in the system on which the NASC can begin to act, can begin to now enforce some regulation rules so as to ensure quality seeds being distributed. At NIHOT, effort is currently ongoing. We have identified materials that meet consumers and farmers' demand, and we are working towards evaluating these materials on multi-locational trial so that we can have these materials nominated for variety release. And then once they are registered and released officially, then NASC can have instruments of office. Then we can develop standards for each of those actors along the seed value chain. Standard procedure, standard operating procedure for each of these actors for their activity, for their seed production. Now, the detailed identification data for the release variety will have to be submitted to both NASC and also to the National Variety Release Committee that is seated in NAGRAB. And we need to have identification of each class of seeds and the source of starter planting materials. For anybody that is working as a nuclear seed producer, where is your source of seed? Where is your starter seed? Where are you getting it from? So NASC, we have all this information and the standards would have been developed that if we are producing nuclear seed, this is the quality of your starting point. This is the quality of your midpoint, and the end line, this is the quality that we expect. The same thing if you are the one producing foundation seeds. Where are you collecting your starter material from? What are the standards operating procedure that you need to follow? What are the things that you need to do? So each of these seed actor is regulated. And then the last thing is that we need awareness and sensitization and benefit of having quality planting materials for plantain and banana if we are to reduce spread of diseases and pests. Next slide. This is the variety testing trial that is ongoing at different locations so that we can have variety release and we can use it as a stepping stone into getting our standards ready for banana and plantain and also to begin the process for regulation and certification. Next slide. Next slide.
Is there?
Yeah, I just briefly put here.
Hello, Dr. Beauty. Hello, Dr. Beauty.
Yes, I'm here with you, Sam.
No, Dr. Beauty. Yes, we have lost her.
Okay.
Dr. Beauty.
She's muted. Maybe she could be advised to unmute.
Okay.
Looks like we're experiencing some network issues.
Hello, sorry.
Yeah. Okay. We can hear you now.
I didn't know I was muted all along.
Okay. Thank you. You can
So
proceed.
we have method. Okay, I can proceed. Okay, methods of getting clean banana and plantain seeds. We can go via the tissue culture, and each of these value chain, this seed actor, if you are producing a nucleus and breeder seed, most times it is best to use the tissue culture. And because the reason this one will be done is because it will be coming from the research station or the private TC labs. So these are the ones that we feed into the other seed actor, like the foundation seed producer and the certified seed producer. So currently there is a project which is ProSiva, the program for seed system innovation for vegetatively propagated crops in Africa, that is championing this seed system, the sustainable development of sustainable seed system for plantain and banana in Nigeria. And also for foundation seed producer, the use of macro propagator, which was mentioned by the first speaker, is a cheaper method for foundation and certified seed producer. And then you can now have those that are nurserymen that will take seeds that have been produced in the macro propagator and begin to establish the nurseries for delivery to different people. So all these people are registered and they are monitored. By the time the system is fully completed and the standards fully developed, Dr. Bakuma is championing that cause, and the standards is well developed and we have released variety, then the issue of mix up in variety for suckers or for seeds will become something that will now be managed and can also be traced. Next slide. So I will not end this presentation without highlighting the importance of using certified seeds. And certified seeds will mean that quality seeds. We know that why do we now have to use the certified seeds? It will give us access to clean seeds, access to varietal purity, access to new genetics, because now we can have newly bred varieties with resilience to different biotic and abiotic stresses. Then we can be guaranteed of the quality assurance. We can be guaranteed of access to premium markets. We can also now be selling seeds within the country and across the country. And most importantly, it will give us access to traceability. If you have bought it from A, then if anything happens, whatever happens on your farm after buying your seed, it can be traced to the source. And this is the beauty of having certification. Aside having clean planting materials, we'll be able to trace the source of the materials, and farmers will have quality for what they are growing. Investors and every other person will have access to quality material, access to premium markets, and they will have new benefits on their investment. Next slide. I want to appreciate your listening time. Thank you very much.
Thank you very much, Dr. Ibitoye, as we learn a very, very good presentation also. This time, we're talking about the certified and clean planting material, which other speakers have already talked about as a means of controlling or managing the disease in terms of the Fusarium TRO4, as well as banana burstop, as well as the other diseases that Dr. Laba mentioned, that's banana wheat. So before we go to the question time, I want to go back a little to see if the national president of the NSPP is still online. If you can give his Google message. If not, I don't know if he's national secretary. Can you give that?
Hello.
Yeah, hello.
Hello,
Yusuf, you're online,
moderator.
sir. Thank you, sir.
Good afternoon.
You have,
Good
sir.
afternoon.
Yes, sir.
I'm online.
Yes, sir. Thank you, sir. You can
Thank
go
you very
ahead.
much. Okay, thank you very much. I'm very sorry because of the network at the beginning. I couldn't join you. So now my goodwill message, please. It is with great pleasure that I welcome all our distinguished speakers, participants, and esteemed colleagues to this very important webinar on plantain stroke banana pestilence, protection against invasive and emerging disease threats in Nigeria. I am truly honored to witness the collaboration between the Nigeria Agricultural Quarantine Services, the International Institute of Tropical Agriculture, IITA, and the NSPP in organizing this crucial event. Plantain and banana crops are lifelines for many Nigerian farmers and play a vital role in our national food security. However, the emergence of new pests and disease threats, such as Fusarium wilt, TR4, and banana bunchy top virus, demands that we remain proactive in finding sustainable solutions. Today's webinar brings together renowned experts such as Dr. Diane Mostad, our very own Dr. Lava Kumar of IITA, and Dr. Bumi Ibitoye, whose invaluable insights certainly enlighten us on current and future strategies to protect these vital crops. I am confident that the knowledge shared today will empower our farmers, researchers, and policymakers to tackle these challenges head on. Let us all take this opportunity to strengthen our commitment to improving plant protection and safeguarding the livelihoods of millions who depend on these crops. Together, we can ensure future where Nigeria continues to thrive in its agricultural endeavors. Once again, I extend my warmest greetings and best wishes to all. Thank you very much, and good luck to everybody. Thank you very much.
Thank you, sir. Thank you for the goodwill message. That was the Professor A. Yusuf, the National President of the Nigerian Society for Plant Protection, which is a very relevant society that has a mandate, has the experts to ensure that our plant material are adequately protected from ravaging pests. So I will use that opportunity to also invite Mr. Idashaba from the Nigeria Agricultural Quarantine Service to give his welcome address. Mr. Idashaba, are you online, sir?
Yes, good afternoon. Confirm you can hear me?
Thank you, sir.
All right. Thank you very much, hosts and participants for putting together this webinar. And then all our invited guests, board members, president, and members of the Nigerian Society for Plant Protection, academicians here present, researchers in the higher institutions and research institutions, all farmers association, and all participants. I welcome you to this webinar. Sorry, I'm coming in late. Had issues with our network, and I want to believe that I can go through this without interruption. And so, on behalf of Nigeria Agricultural Quarantine Service, I thank you for finding time to be here for this webinar, which is organized in conjunction with the Nigerian Society of Plant Protection and preventing the introduction of banana fusarium, wilt, tropical restore, and spread of banana bunchy top disease in Nigeria. As we gather today, we recognize the immense stress that these diseases pose to global and banana production, food security, and the livelihood of millions. Today, we unite to share knowledge, expertise, and best practices in combating this threat. Our collective efforts are vital to safeguarding the world's banana and plantain supply and protecting the interests of farmers, traders, and consumers. This webinar represents a significant step forward in our shared commitment to enhance awareness of yeast disease for monitoring and early reporting, share interests, research and management strategies, promote best practices in the disease detection and surveillance, strengthen phytosanitary measures and quarantine protocols among stakeholders, foster collaborations amongst government, research institutions, and stakeholders, and also to empower farmers and other stakeholders with practical solutions. Let us work together, therefore, to prevent the introduction of Fusarium wilt, tropical rainfall, and eradicate bunchy top disease. Also to protect our plantain and banana diversity and genetic resources. Also to protect smallholder farmers and our local economy. I wish to express my gratitude to our guest speakers, Dr. Diana from Cambridge University, Dr. Lava Kumar from IIT, and Dr. Puni Bitoye from NIFA, Nigerian Society for Plant Protection, our partners in the drive to protect our plant resources, and also our Association of Nigeria for finding time to be part of this collaboration. Your expertise, experience, and dedication will be valuable in shaping effective strategies to combat these diseases. Let us work together, therefore, to safeguard the future of banana plantain production and the well-being of those who depend on it. I thank you very much for putting this together, and hoping that all that we have heard will have helped us to begin to develop plans to contain where necessary and to eradicate where possible. After hearing all this, it becomes a little bit frightening because the feeling will be that maybe in the short while we might not have banana to eat. But God helping us and working very hard, I think we can overcome that. I thank you all for participating. God bless.
Thank you, sir, for the welcome address. I we want everybody also, if you have questions and you have suggestions or comments, you can put it on the chat or raise up your hand. Before the question and answer session, I want to give the floor to Alhaji Dr. Yunus Alidu of the Howard Farmer Association of Nigeria to also give his good message, because we have all Farmers Association of Nigeria on the platform too. We have the members as well as the executive on the platform. So they are mostly concerned about this issue, and I know they will be seriously concerned. So Dr. Yunus Alidu, you have the floor, sir.
Thank you. Good afternoon, all. Confirm everybody's hearing me?
Yes, we can hear you, sir.
Oh, thank you very much. On behalf of the National Executive Council, myself and the All Farmers Association of Nigeria, the Nigerian farmers in general, and I think this is one of the best webinar I have ever had, with the multiple number of people, experts in the fields and academician here. I'm very, very much grateful. And I never expect the meeting is going to be that fruitful like this. And I think with this meeting is a very, very, very big eye-opener for us. Somebody like me, who is a practical farmer and equally a banana farmer, I have never known that some of these diseases exist because we don't put more knowledge on it. And I appreciate everybody who participated, especially those who made presentation, and all your presentation were very, very elaborate. And I think it's very high time we use this information, we disseminate it in the public domain, so that our farmers can know and understand that a banana is not just about putting the sucker on the ground and you expect result. And at the end of the day, this is what is happening. I was so impressed to see that things like this is happening in the country, and it's also coming up. So it's a very good wake-up call for us. So I think I would be grateful if I can plead for your indulgence to share the presentation to us, so that we can also sit down with our farmers and organize another downscaling meeting to disseminate this information to them, especially our farmers in the south, southwest, and southeast, where banana is mostly banana farming is mostly practiced. So I'm very grateful. It's my pleasure. And I wish everybody a successful deliberation. Thank you very much. Thank you very much.
Thank you very much, sir. We will definitely share the presentation as well as the resource person will also be consulted on ways that we can breeze flyers in a format that the farmers can simply understand. So, because of the issue of time, can we indicate by hand raising if you have a question, or you can put it on the platform? We have some limited time. I can take one or two questions. For the three presenters, if it's even three questions. So you can raise up your hand. I'm seeing Alhaji Uruma Abdullahi, please, can you go ahead?
Yeah, good afternoon, ladies and gentlemen. My first question goes to Dr. Diane, who presented on Fusarium with CR4. Part of your solutions to this disease is the use of digital surveillance by FAO with trial in Comoros. If I may ask, apart from Comoros, will it be available for field officers to implement in terms of digital surveillance? Then my next question is on the bungee top banana bungee.
You are muted. We can't hear your second question. Mr. Uruma. Dr. Chris, did you get his second question?
Would the use of tissue culture be appropriate for commercial focus of banana and plantain? These are my questions, please.
I can go ahead and answer the first question. We intentionally developed a digital surveillance tool with the FAO for it because it's quite it doesn't require any subscription fees. So if you are interested, if you're interested to use it, please contact us and we can help you to set it up, because there might be some edits that you want to do that's your country specific, and we can assist with that so that you can conduct surveillance. But definitely it's open to anyone that wants to use it. So you're welcome to contact us about that.
Okay, thank you very much, Dr. Diane.
I would respond to the second question.
Yeah.
See, the soap is only for a fit. It's very effective in terms of preventing aphids from leaving the plant. That is the most important. It's those with wings that can fly when we apply soap water, they no longer can. And also, it is not, the aphid will also die if we use the right dose. Herbicide is very effective. If we use correct we generally use glyphosate, one diluted glyphosate, very, very effective. It eliminates banana mat within 2 weeks. So in terms of using tissue culture plants, of course, you can use tissue culture plants, but you have to make sure that these plants are actually healthy. They are coming from healthy sources. Other means of replanting, such as chopping the infected plants, or they are not very effective, because when you chop, you only chop at the pseudo stems which are above ground. The real stem of banana is underground, and if you want to remove underground stem, it has to be farmer has to dig a pit, and these are stolen like they're very effective, economical, is herbicide injection. Thank you.
Okay, thank you very much, Dr. Olava. I'm not seeing any question again on the chat or anybody.
Hello.
Yeah.
Somebody asked about planting the use of tissue culture material for commercial field establishments.
Hello?
I think somebody asked about that.
Hello?
Yeah. Sorry,
Yes.
I forgot that.
Yes, the source of material from tissue culture can be used to establish commercial field production. However, the person may need to book for these materials way ahead of time. The advantage of using the tissue culture material is that the materials will be the growth will be uniform. The growth will be uniform, and the material you are starting with are very clean and healthy, because tissue culture will not allow any material, any pests and diseases to be multiplied. You can also use the plant materials that come from the macro propagation, because the level of cleanliness and quality of that one follows after the plant tissue culture. So you need to book for these materials way ahead, because
Hello, Dr. Ibutoye. We can't hear her again. Okay, why she...
seedlings are ready.
Okay, we can't hear you initially, Dr. Ibutoye. Okay, let me give other two, and I think that should be the two other participants the access to ask question. Dr. Bushola, can you give you raising up your hand, sir? Can you unmute?
Okay, yeah. Please, can you hear me?
Yeah, we can hear you.
Okay, thank you, sir. I want to commend the three speakers for a very educative presentations. I want to thank Dr. Lavakuma. I want to thank Dr. Ibitoye. Dr. Ibitoye raised a very important point, which is of a big concern, and that's the issue of country and the way it is very easy for diseases of banana to be transmitted. I think that's a very important area that the three bodies here should work on. How do we work on that? And is it possible in developing country where our marketing system is informal, the farming is informal? Is it possible to regulate? Can we have such an internal quarantine system to regulate spreading of this banana disease? So I don't mind if we can have response to that. That's a very big issue, because I know even the National Agricultural Seed Council does not really regulate plant diseases. They work with quality, seed quality and all that. But I think it's the work of this NSPP and quarantine to really, so what can we do in that system? That's my question. Thank you.
Okay, thank you very much.
Thank you very much. Do I respond? Or?
Yeah, you can.
All right, thank you, Dr. Gushola. Thank you very much. Yes, NASC does not regulate pest and disease, but know full well that the standard operating system that is going to be developed for the production of this seed or seeds, they are being developed in conjunction with the protectionists, the agronomists, the breeder. So it is not a one person sitting down to develop the procedure. It means that the procedure is an holistic procedure that we check for pests and diseases along the production chain or along the production line. So once that standard is available, but for banana and plantain, as we speak now, it is not in existence. And there is no instrument of office for NASC to even function. That is for the released variety, for them to run the standards that is going to be developed. It is when we have that registration in place and we have the standard for the seed actors, we have registered seed actors, then we begin to test run it. Because it is not there right now, does not mean that we cannot regulate the markets. But once we need, all we need to do begin to fall into line, into place, then the regulation can begin to happen. And you'll be surprised at the way people will key into it if at the end of the day, it is bringing quality to the table of the entrepreneurs. So it is something that we can do, and it is something that has been done in other vegetatively propagated crops like cassava, happening in yam. People also thought this may not be possible, but as we speak now, it is something that most people have keyed into because now they are seeing good return on the investment. So for banana and plantain, it's something that is possible. We can explore it, but we need to follow each of the steps that we need to follow to get it done and to reach the place where we are going.
Okay.
Thank you.
Thank you very much, Dr. Evitoye, for that response. We have, I think probably that would be the last one because of the issue of time. Mr. Ambrose Chineke, can you unmute yourself, sir?
Good day, everybody. I want to thank all the speakers. We have learned a lot of the diseases and also the certification. My question, first of all, goes to Dr. Lavar. You gave a very great presentation. But I'm wondering if this banana bunch top has been found in an enclave for over 11 years and is still there. Is it that what measures are being done to either contain or eradicate? What are the obstacles? Yes, you mentioned the root system, the sucker system is very huge, require regulation. Is it the only problem? Then concerning seed certification, I know NIHOT is your mandate. You have the core competence as far as varieties of plantain. So what effort is NIHOT doing, to collaborate with the NASC? Actually, NASC, yes, oversees all the seed system. But since it's your mandate, have you developed a system where we can have all the varieties, we can easily identify it, and say, okay, this is a traceable, this particular variety, these are characteristics, because they are very, very important in developing that standards we are talking about. So that then individuals, those seed participants, stakeholders, can they come to you and say, yes, you have all the varieties lined up and easily identifiable? That's my question.
Okay, thank you, sir. Dr. Lava.
Yeah, thank you. Thank you for the question, and nice to hear from you, Dr. Ambrose. Yes, there have been measures to contain the spread from those 3 local government areas. We often cite it as a kind of a limited success story for the reason that those efforts have prevented the virus spread from that particular location to other parts of Nigeria. The same could not be achieved in a number of countries. This is of many reasons, but the most important being the presence of quarantine in that area. Since it is close to border, there are many checkpoints, better awareness, and all of it. Why we could not eradicate even 10 years? It's very difficult problem to eradicate. It requires farmers' engagement, full commitment by the farmers to participate. We have done continuous programs jointly with NIHOT through multiple projects for nearly 6 years, working with communities in terms of encouraging them to eradicate plants and then supply clean planting material for replanting. But for as explained, banana being banana as it is grown mixed crop, or there are orphan plants, we establish new fields, then there are still infected sources around these new fields getting infected. Not all farmers are participating in eradication program because for various reasons. So this is a challenge. If we want to eradicate, eradication is only possible in a very short window when the infection is in one or two places in a small communities. Then we can voluntarily go and take action. When it is widespread, the 3 local government areas where the disease is spread is equivalent to the country of size Burundi. It's quite big, right? It's quite big. So, and there are many farms infected. It is possible. I'm not saying it is impossible, but it is possible, but it requires persistent commitment from the implementers such as us, as well as farmers. And it is also slightly expensive. And if both farmers and we participate, we train them, farmers take it voluntarily to eradicate, and we supply as incentive planting material, then they do the local monitoring and all, then it becomes easier. So it is definitely more challenging in a situation where the communities have multiple avocations. So the farmers get distracted. As I mentioned, it requires long-term focus. It is not something we can eradicate in one go. It requires long-term focus. So this experience is not unique only here. I can also cite the same situation in Australia. Australia is using nearly annually, they are investing 200 million. No, not 200 million. It's quite substantial amount of budget to eradicate banana bunch top. And this is happening for more than 40 years. And they have been very successful in avoiding the disease coming into commercial fields, but full eradication was not possible. If they withdraw monitoring and surveillance, the disease is reappearing in the commercial plantations. So this shows the challenge. Even in an intensively managed situation, this disease eradication is difficult, and that is the reason why it is considered as among the top 100 countries. I mean, top 100 invasive species. So we need to do more, and our current projects have exhausted. So now our activities have reduced, but we are still focusing on awareness raising and trying to do our best. But yeah, it requires government intervention. So we can talk further on this some other time. Thank you.
Thank you.
It's a challenging problem.
Thank
Yeah,
you
but
so much.
we are definitely working along with quarantine agency.
Thank you, Dr. Lava. We continue working in collaboration to assist to ensure that this disease is eradicated or the spread is limited. Dr. Ibitoye, we have the next question. Are you there? Dr. Ibitoye? Okay. I think, okay, probably she's not there. Let me just answer briefly for her. She mentioned in her presentation that they're having a project currently because at the moment the informal sector is the one that is um, selling plantain materials. So now they have a current project that is ongoing to ensure that banana or plantain is now formalized in this a sector that it is registered and the seed are certified in such a way that we have clean planting materials that we know that is clean from all this disease. Apart from disease, is the quality is assured. So they are working in collaboration with the Seed Council as well as the other relevant like IIT and relevant bodies to ensure that these plant materials are easily available for farmers. Okay, Dr. Vito is here. So you want to respond, ma'am?
Yes, I'm so sorry. The network, because I'm in transit, so the network has been up and down.
I understand. Thank you.
Just thank you. Yeah, you have given the insight that I've talked about the Prosiva project, which is being led by IIT, and a lot, not only banana and plantain, but other vegetatively propagated crop like cassava, potato, and yam are also crops that are being considered in the project. However, what makes banana a bit unique is because banana is the only crop among all these VPCs that have not had any registered variety in the country, so to say. But maybe for East Africa, they have registered banana varieties. But in Nigeria, and I think Ghana, there is none existing. So this is what we are doing together with NASC to actually, first of all, develop the standard procedures for the different actors along the seed value chain. Once that is done, we are doing that aside, and then separately, we are working towards registration of good varieties of plantain and banana that meets stakeholders' demands. Because we all know the place of plantain in our culinary diet in Nigeria. A crop like plantain has its own specific traits that consumer prefer. And so if we are to bring in even improved varieties, the improved varieties must meet this existing standard.
She's traveling. She can just where there's network.
Okay, you're off, Dr. Ibitui. Dr. Ibitui. Okay, she actually is in transit now because she went to she she's in transit. So we would want to end the program because of the issue of time. We want to first of all say that the presentation will be available on different platforms as well as the recording so that we can go over it. Then we also have opportunity for collaboration as research in terms of the different pests that were listed. So one of the issue we need to continue doing is the issue of surveillance. And like Dr. Lava and Dr. Diane talked about the issue of the digital world now. So we're trying to develop a platform where this pest will be reported. Yeah, I know you have
that
the connection is gone. Dr. Bito, don't worry. We'll continue. So we'll have opportunity like Dr. Diane said, they said that we have is one of the Fusarium wheat species. So it's an opportunity for researchers to see if they can go around and see if they have symptoms. Then we will assist in terms of documentation to send samples out to Stellenbosch University if you have samples you want to send for confirmation. But you need to report that disease, and then let's see if it's actually the one that has that, if it has that symptoms. So we can now send confirmation to Stellenbosch University. We assist in that regard any researcher that is interested. So we want to collaborate in that terms. So when the reporting platform for various diseases is ready, we are developing one along with Dr. Lava in the IT. So once ready, we'll also put it in the platform for us to report any type of disease that we're seeing in the country. So that way we can easily conduct our surveillance. Then also, I want to use this opportunity to thank the speakers, Dr. Diane. She's going for that meeting presently because we have exceeded the time frame. Then Dr. Lava Kuma, we appreciate you. And Dr. Bumi Bitoyi, we also appreciate her. She took time because she took her daughter to the university, so she's on transit back again. So we thank you. Then we want to use the opportunity to also thank the board member of the Nigerian Society for Plant Protection. They have been our partners, and then they have collaborated with us to ensure that this webinar takes place. So we are also very, very thankful to the executive of the Nigerian Society for Plant Protection, most especially the president, Professor A. Yusuf, for championing the webinar will have to take place. So I'm thanking you, sir, for this opportunity, as well as the ESCOs members too. I'm seeing a lot of them on the platform. Then I'm also using the opportunity to thank the All Farmer Association of Nigeria for their support. We'll continue to work together to ensure that this disease does not come into Nigeria. and then the BBTB does not spread. So thank you, sir. And also we want to thank the officers of the Nigerian Agricultural Quarantine Service. I'm seeing them also on the platform. So we want to thank everybody. If I've not recognized you, please pardon me. I'm also thanking all the participants. I assure you that we will send out the link to the recording as well as the presentation to the various platform where we used to announce the webinar. So I'm also seeing somebody from Ghana. I will note that, and then we will communicate also. The Plant Protection Service of Ghana, we want to also collaborate with you so that we work together in the continent to ensure that we don't have the disease. So I will communicate with you there on that. So thank you for everybody. Thank you for your time. Thank you for your patience. We will have this another time.